Myself Sucheta Bidve, and today we are going to continue our second part from the seed dormancy. Student, in last lecture we have already discussed about the what is seed dormancy and different types of seed dormancy. In today's lecture we are going to discuss reasons of seed dormancy and methods to overcome the seed dormancy. Let's have a look on causes of seed dormancy. Student, seed dormancy is mainly induced by seed coat. We know what is seed coat? Seed coat is integumentary layer of the ovule. Okay, during formation of seed, there is deposition of number of polysaccharides, hemicellulose, wax, fats, etc. within this integumentary layer. And at the time of seed ripening, this component get dehydrated and forms hard and tough seed coat. And presence of this tough seed coat is one of the important reason of the seed dormancy. Now, this seed coat induced dormancy may be caused by three different ways. First one is seed coat induced dormancy caused due to water impermeability. Due to presence of tough seed coat, the water couldn't enter within the seed molecule. And within we know that for the germination of seed, the absorption of water by seed is very important. For the process of germination, initially water is get absorbed by the seed and it leads to imbibition pressure development within the seed, which leads to metabolization of different metabolite which are present within the seed, which is the water is very important for the first step of the seed germination. But here, due to presence of tough seed coat, there is no permeability for the water within the seed. And as there is no entry of water molecule within the seed remain dormant as it is. This type of dormant seed are mainly found in the family Leguminosae, Nymphaceae, and Solanaceae. You might be know that the seeds of lotus they are require a very long period for the germination due to presence of very tough seed coat. The second cause is gas impermeability. As the seed coat is very tough, there is no exchange of gases. As there is no exchange of gases, the oxygen is unavailable for the internal metabolite and so that the germination doesn't occur. And this type of dormancy is mainly found in the seeds of xanthium. Nextly, the mechanical resistance of seed coat to the growth of embryo. Now, in this category of seed dormancy, mainly it caused due to presence of very tough seed coat, and this tough seed coat is resisting expansion of embryo. It suppresses the development of embryo, and due to presence of undeveloped embryo, there is no germination of seed. But still, this type of dormancy doesn't remain for very longer time. Within a short period of time, the seed coat can be broken by internal embryo and there is seed germination. This type of seed dormancy is mainly found in the plant Amaranthus and Capsella. So, this is the first important cause of the seed dormancy due to presence of tough seed coat. And due to this tough seed coat, there is water impermeability, there is gas impermeability which resists the germination of seed. And finally, this tough seed coat create the mechanical resistance to the seed coat due to which there is no growth of embryo and so there is no seed germination. So this is the first cause of seed dormancy. Nextly, The second type of seed dormancy is mainly found due to development or due to condition of embryo. Such a type of dormancies are mainly embryo-induced dormancy. See, remember, there are two main causes of dormancy. First is tough seed coat and second one is embryo-induced dormancy. Now, this embryo-induced dormancy can be caused due to rudimentary or poorly developed embryo, or it can be caused by unresumed growth of the embryo. We'll see one by one. First one is rudimentary or poorly developed embryo. In this case, the embryo does not develop rapidly as compared to surrounding tissue. Okay, so endosperm tissue will get grown rapidly and the embryo remain as it is undeveloped, and due to presence of this undeveloped embryo there will be seed dormancy. But this type of dormancy can be recovered within a very short period of the rest. Okay, after a period of rest, this type of embryo get fully developed and get germinated into new plant. And this type of dormancy is mainly found within the family Orchidaceae. Orchid, we know, everybody knows the plant orchid. Within this Orchidaceae family, the embryo remain undeveloped in initial stages of seed development, but after complete growth of the seed, the embryo undergoes the developmental stages, it completes its development and then it get germinated. Likewise, same type of dormancy is found in Ginkgo biloba also. Again, Ginkgo biloba, it is a gymnospermic plant. The next cause of embryo-induced dormancy is embryo is fully developed but unable to resume growth. This type of dormancy is mainly caused due to unfavorable environmental conditions. Or in the same type of dormancy, the seed is not going to germinate even if the favorable conditions of the seeds are of environment are present. If we remove the seed coat of this type of seed, then also this type of seeds are failed to germinate. In this condition, mainly the embryo condition of embryo is not developed or not completed up to the germination. Due to physiological condition of seed, it is unable to achieve its growth. In this type of dormancy, germination can be regained by a longer cold storage. And this type of dormancy is mainly found in the apple, peach, cherry, etc. Okay, so these are the two important causes of the seed dormancy. Now, as these are the causes of seed dormancy, there are number of methods are available to break the seed dormancy. Now, within these, we'll see one by one. Within these, first one, very easiest method is nicking. Nicking, simply taking a cut at the distal end of the seed with the help of sharp tool. Okay, the distal end, generally in a hard seed coat type of seeds, we can take a simple cut within the seed at the distal end, and this type of seeds are allowed to germinate. But practically, it is applicable for only small quantity of seed, as well as the seed must be enough large to take cut at the distal end. So, this type of method can be applicable in the seeds like acacia, albizia, dolonex, etc. Within this plant, we can cut the seed at the distal end and we can achieve the germination. The next method, which is the very simple most method, and it can be applied to number of seeds it can be easily applied without any expenditure, and this method is soaking of seed within the water. Within this, the seeds are soaked within the water from 2 to 48 hours, which leads to germination. What happened? Due to soaking in the water molecule get entered within the seed and it leads to activation of enzyme and reserve metabolites which are present within the seed, which leads to softening of seed coat and leaching out of inhibitors which are present within the seeds, and it leads to germination of seed. This type of this method is practically applicable within the seeds of acacia, albizia, pinus. Mostly in all tree species, we can easily apply this method to break a seed dormancy. The next one is the, the plant or the seeds in which simple water soaking is not enough. In that cases, we are using hot water soaking. Now within this hot water soaking, the seeds with a hard seed coat are used for the soaking, and generally the temperature of water is ranging from 40 to 100 degrees centigrade for the specific period of the time. And after soaking, this soaking period is depend upon the hardness of the seed coat, and after soaking the seeds are allowed to germinate. This is the quickest, cheapest and simple method which is whose requirement are very less. The example within the Tamarindus, Adansonia, Calendra species, this water soaking method can be practically applied. The next method for the species and specifically whose seed coat is very tough, and on the seed coat of such a plant seed, there is no use of soaking in water or hot water soaking. In that cases, this acid scarification method is used. Within this acid scarification, simply the seeds are soaked within the 95% pure sulfuric acid. Due to this soaking is this soaking treatment is given for the different period of time with from it start from few hours to 1 to 2 hours to up to 24 hours. Within this period of soaking within the acid, there is softening of seed coat, and this softened seed coat are then allowed for the germination. But this method is not very common because the use of concentrated sulfuric acid, it has some risk point and again the problem of cost, so that this method is not practically applicable for all the plants. But in specifically tree species like Melina arborea, this type of acid scarification is applied for the seed germination. The next method is mechanical scarification. Now simply the mechanical breaking of seed coat to weaken the seed coat. This method is applicable, the breaking of seed coat. But again, this can be applicable for very small quantity of seed, and the seed size should be enough larger in order to break the mechanically seed coat. This method is almost applicable in all tree species. The next method is mainly used for the embryo caused seed dormancy. Okay, the next method is stratification, and this method is mainly implemented for the seed dormancy caused by the embryo. In this case, or in this type of treatment, seeds are exposed to well aerated, moist condition at low temperature. The range of temperature may vary from 0 degree centigrade to 10 degree centigrade, and the treatment time may varies from few hours to few weeks to few months, and it leads to breakdown of physiological dormancy. If you remember, when I taught you the physiological dormancy, at that time I told you the breaking of physiological type of dormancy is possible only by stratification type of treatment, in which the seeds are exposed to the low temperature from few hours to few months, and this type of treatments are effective in the dormant seeds of Castanopsis species or Cucurbit species. In this type, there is utilization as well as this type of method are mainly applicable in Acer species, in which the physiological dormancy is mainly present, and the breaking of this physiological type of dormancy is achieved by means of stratification. And finally, the simple most method which can be widely applied to the all type of agricultural crops, and it can be applicable to plenty of seeds, to all the cereal crops, to all the pulses crop, and this method is utilization of PGRs. PGRs, plant growth regulators. The utilization of these plant growth regulators in order to break the seed dormancy is most and simple and practical method to achieve the seed germination. In this method, simply the gibberellic acid is used for the breaking of seed dormancy, and again it improves the rate of germination in the agricultural crop. The application of GA3 may be through foliar spray or by giving treatment of GA3 to the seed directly. The different concentration of GA3 can be applicable, which depends upon the type of plant species and type and structure of seed. So simply by applying GA3 to the agricultural seed, we can achieve the germination properly, and we can increase the rate of germination into the agricultural crop. So these seven are the major methods of breaking seed dormancy into the economically important crops. These are some references which I have used for our today's lecture. You can simply find out this literature in any physiological book also. So, thank you so much. If you have any query, you can find me on my mail, or we can talk on our Google Classroom also. Okay, thank you.